you go to put the oil in.
Alrighty.
Now, let's do that one more time so you don't screw up. You're gonna have to lean up your strip, fill every cup halfway to the base of the cup. Except for these three, you're gonna fill all the way with your unknown emulsion. Then, you're gonna lay your strip down flat in the base, get a new pipette, and fill these, the top half, with oil. Okay? Now, one part I didn't tell you about that you're going to have to do before is when you take your base, see it has all these little cups in it? Take your squeezy bottle of water and you want to fill all those little cuppies. But you don't want to overflow them. You want the water in the cups, not over the surface. Good luck with that. The reason is you don't want your strip to dry out in the incubator. So the water doesn't have to be sterile or deionized, it doesn't matter, it's just moist. So it's moisture, so when you're incubating, your strip doesn't dry out. Okay? So probably you want to do that second. What's the first thing you're going to do? Label your strip. So you can write your team name or your unknown number on this little flap. Then put your base down, fill the little cuppies with water. Then you're going to fill your cups halfway with emulsion, fill the three full way with the emulsion. For the green ones, go back and fill them with oil by laying it down and then put the oil in, okay? Then you're going to put your cover on. And then you're going to place it in that bin over there by the sink. And when everybody's are done, I'm going to stick them in the incubator. Whoever's leaving the room is the last one to go. Remind me to put the bin in the incubator. Sometimes I forget, okay? Okay. We're going to incubate them 24 hours. Tomorrow we're going to come back and we're going to work on our scorecard. On your scorecard, you'll see little numbers in each little cup. If you get a positive reaction, you're going to circle the little number for each little cup. If you get a negative, no color change, don't circle it. Notice how the little cups are in little boxes with an oval at the bottom. When you're done deciding, you're going to add up, let's say the first box, you circled the 1, and you didn't circle the next 2. That's the end of the box. You have to add up the numbers you circled and put it in the oval. You're going to end up with a 7-digit code that we're going to feed into the computer tomorrow, and the computer will tell us what your unknown is and the likelihood, the percentage likelihood that that's what it is. So what they do is they have a database of all the different strains of Serratia, all the different strains of E. coli, all the different strains of Salmonella, and whatever else. And they're going to compare it statistically. It's 99% similar to all the other Salmonella enterotitis strains that we have. Or maybe it's only 50% similar to all the other E. coli strains that we have in the database. 50% is not good. 80% is pretty good. That means the likeliness that your unknown is correctly identified and not contaminated. If you have contamination in your unknown, it's going to affect your results a little bit, and you might get a lower percentage. Okay. In your API test strip, it's called an API 20E. You need to know this for your last quiz. API stands for Analytical Profile Index. If you read your protocol, you'll see it. So it literally profiles bacteria. And then you're going to feed in your 7-digit code, and it's going to tell you if your profile fits the database, the profiles for others of the same species. And the 20E means it's 20 tests for differentiating Enterobacteriaceae. That's what it stands for. There are also API test strips for diagnosing staphylococcus and that kind of stuff, all kinds of different bacteria. All right. So you know how to do that. There's two other tests that you can do that are not going to have any bearing on your thing that you're doing, but they're just fun to do. All right. In your scorecard, notice the last, well, there's a space. So see, here's all your little tests that you're going to do, and then there's a little space and another box, right? You don't have those on your test strip. Okay? So we're not going to worry about those. But you also, the oxidase test here, see how it's got a dotted line? That's because it's actually a separate test. It's not in the strip either. Your test actually ends at ARA. That's where your strip ends. The oxidase test has got a dotted line because it's a separate test that we can do. But we don't even need to do it because I'm telling you right now, remember this for your lab quiz. All Enterobacteriaceae are oxidase negative. Oxidase negative. But just for kicks and giggles, if you want to, you can do an oxidase test up here. I'll show you in a minute. There's another test, isn't even on your API test strip. It's a catalase test. For the most part, Enterobacteriaceae are catalase positive, which means they can break down hydrogen peroxide. Hydrogen peroxide is an oxidizing agent. It burns, just like you burn your glucose. It burns.
Can break down what?
Huh?
Hydrogen?
Peroxide.
Sure.
It's hydrogen peroxide. You know the brown bottle at the drugstore? It's the same crap. Buy it at the drugstore, put it in a little bottle for you. If the bacteria are catalase positive, that means they have catalase enzyme that helps them break hydrogen peroxide down into water and oxygen. So you're going to get bubbling. Bacteria that can do aerobic metabolism. So that includes your aerobes and your facultative anaerobes. Typically are catalase positive. It's the anaerobes who are catalase negative. So all your Enterobacteriaceae are oxidase negative, catalase positive. I had boo-boo on my finger because like a